Alright students, as Dr. Gruel working on the end of chapter 3 here. I'm going to post here just some information from where we left off to the end of chapter 3, and then I'll post another link to a small video about Hox genes in the homeobox and homeodomain, and we'll have a short quiz on these on Wednesday, which is tomorrow. I'm going to try and keep this to 10 minutes or less. The other video is about 10 minutes, so shouldn't be too much. That'll help us wrap up chapter 3 and move on, so we can get one more chapter knocked out before the test, which is a week from Friday. I'm just going to slowly go through some of these slides. I had made some changes, and they weren't present there, and I just want you to have access to that, so you can just pause this as you go through on YouTube or wherever. So remember that DLSs are chromatids and homologous chromosomes and how those are different. We talked about location and genetic map. I don't think there's anything different there. I think pretty much here was the same. We're just talking about what happens if you change one gene, and really the take-home message is between bacteria, invertebrates like worms and flies, and humans, there's a lot of different things that can happen as you manipulate a single gene. Okay, the probability of the single gene base change has a functional consequence is low, then where does the diversity of natural selection come from? The basic idea here is changing one base pair has very little effect on the overall genome, especially after the organism is born. It's almost impossible to have an effect because of the division between the germline and the somatic cells. Any changes that happen in the somatic cells can't be passed on to the next generation. It has to be in the germline, has to be in the line of cells making the gametes. But as we see throughout evolutionary history, there's many mutations, many locations, many individuals, long periods of time. All this put together can cause significant changes in DNA of organisms evolving over time, and that can add up to what we see in evolution. It's good that it doesn't happen more quickly, and it's good that a single base pair change can have very little effect, because basically it would result in death all the time with the occasional positive mutation. So we see mutations accrue at all sites in the genome and all cells, but most mutations have little effect on the function of the cell or the organism, and are mostly expected to be selectively neutral. They're also somatic, so even when you have the change, when you have lethal mutations, if they happen in somatic cells, usually the cells die. It does not get passed on to the next generation. I'm going to speed up here. Redundancy and robustness. And redundancy, we're basically dealing with paralogs and alleles. I think I mentioned that, but just to make that clear, we really want to focus on here the paralogs and the alleles. Robustness is an important concept. I think redundancy is a far more important concept, happens a lot more often. Alright, and then alternative splicing to make multiple gene products. And like I think I mentioned in class, 22,000 genes, 35,000 proteins versus 21,000 genes and 100,000 proteins in Homo sapiens. Big difference there. Alright. Okay, for time's sake, I jumped a few slides here to the CNV discussion, copy number variation. Like I said, copy number variation can be a few bases and can be up to 1,000 base pairs and a lot more than that. Our book is going to call CNVs 1,000 bases or more. And those can be repeated many, many times. And so we get changes in the number of copies of things through copy number variation. And you can see here, it turns out to be an extremely important principle, because between humans and chimps, we have about 400,000 changes in protein coding regions. We have 800,000 to 1 million changes in copy number variation. Very, very important concept in delimiting species, so it appears at this point as we're looking at it. That takes us to a concept of gene families. Gene families are genes within the genome that have readily identifiable sequence similarity, and they carry out very similar functions. So again, we're back on this subject of paralogs. Inside the organism, we have genes that are duplicated, and then they can be co-opted to have different functions. So if you have two genes that have the exact same function, then when one mutates, it doesn't cause death of the organism. It may cause a new gene to be born, or may cause what we call a pseudogene. Sometimes one of those genes just dies, and that's a pseudogene at that point. It doesn't code for anything. A really cool example of this that we've seen in just the evolution of humans. In humans living in areas of high starch diets, the average number of amylase genes turns out to be slightly higher than those in low starch diet areas, because amylase helps process that starch, especially in the saliva, start to process it. So a very neat little take-home message from there. Here's just an example of what might happen historically. So we have our ancestral gene up here. The gene gets duplicated. The further duplication and divergence in function, we make all these different genes. As they become duplicated, they can have different functions. We get things like pseudogenes. Pseudogenes is when that gene dies. It doesn't have any functionality anymore. A good example of that over here is the myoglobin. Myoglobin is an ancient gene that we all had. Helps with oxygen in muscles. And it's still preserved. In chromosome 22 of humans, we see that myoglobin still as a functional gene because it is so important. We can't do without it. But the gene has been copied and has given us this alpha globins and beta globins and all sorts of those that are expressed at different points in time. They're expressed in different tissues. This gene family becomes a whole bunch of different genes that are actually quite similar to each other, doing similar things. All of them are dealing with oxygen. A lot of these others are in the blood and other places like that. But they're very, very beneficial. An interesting case of this in the FAD2 genes in coffee. Coffee has two characteristics that people really like in their coffee. That is the aroma and the amount of caffeine present in the coffee. And this CCFAD2 genes have all been copied. So there's 6 copies of that gene, and they're expressed in different parts of that coffee plant in different numbers. Some of them are expressed in huge numbers, and there's lots of these genes, and they're expressed in huge numbers for these great aromatic qualities. You can see that FAD2 in Arabidopsis, it exists there. It's not expressed really in any of those tissues in Arabidopsis because no one really cares how their Arabidopsis smells, believe it or not. All right, this is a link. I'm going to post this link in the email. This is to that to the Hox gene YouTube thing that they have for the textbook. I think it's very helpful and useful in looking at. So I'm going to have you kind of do the Hox genes just through that on your own. So just these last few things are kind of just as a review. Major sources of genome differences are base substitutions, insertions, deletions, changes in copies of genes, and then a differential expression of copies in different genes. I'm gonna come back to that. This figure is one that I really, really like because it talks about all these different changes, genetic changes we see between species and how common those are between how distantly related these organisms are. So as we look at here, individuals in the same species, the amount of phenotypic variation comes from gene number differences, regulatory sequences, and variation in amino acids. Closely related species, now we're going to throw in the occasional novel gene. Remember, we talked about humans, chimps have only 150 to 200 genes different between humans and chimps. Chimps, variation in splicing, right? That's going to deal with different proteins we can make out of the same sets of genes. And then, of course, variation in gene number, again, regulatory sequences, amino acids. Then we get distinct related species. Those bars just spread out a little further, right? And I'm going to go back to here. This is just, whoa, didn't work. Alright, going back to this slide, I could sit here and read it for you, but I just encourage you to spend some time with it. This is a great little summary slide of where we get the phenotypic and morphological changes, effects from the genetic variation, right? Variation amino acid sequences, gene regulation, splicing, novel genes, and RNA encoding genes. It's a great just summary slide to look at. Here's an example of gene regulation resulting in phenotypic variation, causing change in hair color, depending on the amount of expression. All of that coming back to one single base pair change in a regulatory gene, 350 kilobases away, 350,000 base pairs away from that gene, that KIT ligand gene, changes its expression, changes your hair color. Pretty cool stuff. And I think we've already talked about this plenty. So I'm going to sign off here. Went a little over 11 minutes. That's okay. And then I'll also include the other thing to watch. It's about a 10-minute thing. And prepare for a quiz on these two things. It won't be highly detailed. It'll be more to test, did you actually watch it or not, on Wednesday. Thank you very much. Oops.